السلام عليكم. Dear participants, I would like to thank you for your participation and sharing your research work to the first international conference on environmental biotechnology and biodiversity. I'm Dr. Hamid Sonia, and I'm a member from Europe. With us, Mr. Bujna, moderator of this session from ICEBB. Mr. Bujna, are you here? Yes. Mr. Bujna.
Mr. Bujna, please. Okay, sorry. Mr. Bujna, good morning, good afternoon, sorry. Mr. Bujna, please enable your microphone. If you are here, please raise your hand or write something by SMS. Dr. Bujna, the moderator of this session. Okay.
Oui, bonjour.
Bonjour.
Oui, bonjour.
Oui. Okay, just I will reply to Amina Tahiri. It's room 1. Okay.
Oui.
Okay, let's start. I think we have a good internet connection. Okay. Okay.
La connexion n'est pas bonne. Ici, à Bousmaïn, on a des problèmes un peu avec la connexion. Vous m'entendez?
Okay, no problem. So oui, on vous entend très bien.
Okay, so before we start this session, I will agree on the project. I will share your presentation, one in my screen, one by one, according to the ID number. After each presentation, you can give your question or comment. So, are you ready? Okay. We start by communication number 38. We have at 5. The matter to receive. Okay. So I program. Okay, so I share my screen for first communication.
Okay, so the first the first presentation by Dr. Hamid. INV38 and the title Zooplankton Associations and Environmental Factors in the Shifa Wadi, North Central Algeria.
I would first like to thank you for your interest in my work. It is an honor for me to have the opportunity to present the result of my research entitled Ecological Investigation of Zooplankton Abundance in the Shifa Wadi, North Central Algeria, Impact of Environmental Variables. Currently, aquatic ecosystems, especially rivers, suffer from increasing degradation of water quality, with the path more rapid due to various human activities in the watershed. The unwanted influx of agricultural and industrial waste into these lakes affects their physicochemical and biological composition. The aim of our study is to investigate the special composition and community structure of the Media Mountains and sink in the bottom of the gorge of Shifa, in the Lydian Atlas between the Blida Massif and the Mosaic Massif. With a length of 35 kilometers, it is one of the main tributaries to Mazafran River. Shifa Wadi is located in the northwestern part of Algeria, Figure 1. It originates in the Media Mountains and sink in the bottom of the gorge of Shifa, in the Lydian Atlas between the Blida Massif and the Mosaic Massif. With a length of 35 kilometers, it is one of the main tributaries to Mazafran River. The surface water samples were collected from 6 points at Shifa Wadi, Figure 2, from June to August 2019. At each site, pH, salinity, electrical conductivity, water temperature, and dissolved oxygen were measured in situ with a WTW multiline 34i portable meter. The results of mean value of physico-chemical parameters for each station are given in Table 1. The water temperature in this fluctuated between 21 Celsius and 26 S4 and S3 respectively. Dissolved oxygen concentration showed a trend varying from 3.24 to 6.48 mg per liter. Human activities, and mainly those related to agriculture, are major cause of the presence of nitrate, and phosphorus in surface water. In this study, the nitrate concentration in the range from 3.02 to 12.8 mg per liter. The highest concentration of PO4 was recorded at Site 3, 5.92 mg per liter. The main environmental impact associated with phosphate pollution is eutrophication. 21 species of zooplankton were identified from the Shifa River. They belong to Rotifera, 15 species, Cladophora, 4 species, and Copepoda, 2 species. The Rotifera constitute the largest group, making up 72.24% of the zooplankton population, with organisms ranging between 110 to 618 organisms per meter cube. This was followed by Cladocera, 16.98%, and Copepoda, 10.94%, which had a density between 0 to 110 organisms per cub meter. The variation in dominance, diversity, evenness, and richness of community structure are given in Table 2. The species number value for the 6 sites were from 9 to 20 species. Therefore, the value was lowest for the most polluted sampling point, S3, and highest for the upstream point. The Shannon Diversity Index appeared to be an efficient tool to evaluate the structural complexity of the plankton assemblage. A low value of diversity was observed in S3, and certainly it is related to different factors, pollution and eutrophication. For environment factor impact, results of FDA analysis show that it is possible to distinguish 2 groups of factors which predicted spatial distribution of biological variables. The first group of factors included nitrate, phosphate, and water temperature, and they were connected with the stations 1, 2, and 6. The second group included dissolved oxygen and electrical conductivity, which were associated with station 4. S2 is very far from other stations. The RDA figure demonstrates nitrate, phosphate, WT, and pH were the main predictors of distribution of zooplankton abundance. Most zooplankton species are grouped in S2. They had a negative correlation with the previous factors. Brachios angularis was related to higher value of dissolved oxygen. This result may indicate an indirect relationship of these species with phytoplankton biomass, because a higher value of dissolved oxygen indicate higher primary production. The influence of physico-chemical properties of Shifa Wadi on its zooplankton composition and abundance were investigated at 6 sites in summer 2019. The present study records for the first time the aspect of zooplankton diversity and composition in relation to the physico-chemical environment of the Shifa Wadi. Results show that the structural variable of the zooplankton of the Shifa Wadi indicate pronounced eutrophication of its ecosystem. The dominance of eutrophic species at this period proved especially rotifer species. The alkaline pH, food abundance, and nutrients were some of the factors that could limit zooplankton growth, composition, and abundance in the aquatic ecosystem. Thank you.
Thank you so much for your presentation. And I ask if you have any question or comment. Yes, Amina Tahri.
Good afternoon. Je peux poser la question en français si vous pouvez.
Il n'y a pas de problème.
Oui.
Oui, bien sûr.
Bon, c'était très intéressant votre présentation. Je voulais savoir seulement, vous avez mesuré la conductivité électrique. Ça devrait être mieux de calculer la salinité. Vous pouvez, de la conductivité électrique, il y a un tableau de conversion pour la rendre comme étant la salinité. Ça te donne plus, c'est beaucoup plus significatif dans vos résultats. La deuxième question, je voulais savoir, c'est quel logiciel que vous avez utilisé pour l'étude statistique? Et merci.
Merci Amina. Donc pour la salinité et la conductivité, généralement on mesure la salinité et la conductivité, sauf quand on fait l'étude statistique et on présente les résultats dans RDA ou la CPCA. Généralement, pour avoir des corrélations significatives, on élimine des paramètres. Et dans cette présentation, donc, j'ai éliminé la salinité. Alors, pour l'analyse statistique, donc, généralement, on utilise le logiciel R. Donc, voilà, c'est le logiciel R.
D'accord, merci.
Voilà.
Further question? So if you have any question, you can write an email to Dr. Sumia Hamil. So thank you for Sumia Hamil for the nice presentation. The next presentation is given by Shafa'i Amin, ID 53. Shafa'i Amin, are you here?
Oui, je t'entends très bien. Vas-y.
Okay, thank you so much.
You're welcome.
Okay.
kilometers from the capital and 25 kilometers from the Mediterranean. Good morning, everyone. My name is Amin Shafai. I am a teacher researcher from the University of Science and Technology, Houari Boumediene, Algeria. Firstly, I would like to thank the team of International Conference on Environment, Biotechnology and Biodiversity to accept my participation, and it's my great pleasure and honor to share with you a small part of my research entitled Diversity of Phytoplankton Population in the Hamiz Lake. As we know, water is life, and phytoplankton is part of this life, simply because phytoplankton are at the base of aquatic food webs. And many of these species have a global importance for ecosystem function and service. Sadly, studies on this topic in limnic system under Mediterranean climate have been rarely estimated, especially in Algeria, where it is limited to a few areas like river and basin. It is only these last few years that several research have been undertaken on various sites. At the same time, the work devoted to various aspects of phytoplankton ecology in them has diversified, while it is still limited. For that, the goal with this study is firstly to characterize the algal population that live in the Hamiz Lake and to analyze the temporal variation of phytoplankton. Then to evaluate the variation of physico-chemical parameters of water, and finally to understand the multiple factors which drive ecological change in phytoplankton community. And because of the constraint of time, we're gonna share with you this time only the first two objectives of our study. Hamiz Reservoir is one of the eldest reservoirs in Algeria, situated in the area of Wadi 14 in the lower of Kabylie of Georgia. The site is located 35 kilometers from the capital and 25 kilometers from the Mediterranean Sea. Also, the reservoir is mostly used for irrigation of the Mitija. Times provide the source of drinking water. For sampling, samples of water were collected monthly over a year and analyzed for nutrient content and phytoplankton density. Two types of phytoplankton sample was made. Horizontal sampling for qualitative study and punctual sampling for quantitative study. Both samples were stored by formal at 5%. For the analysis of phytoplankton in the laboratory, taxa were identified using light microscopic cases like Burley and Kramer. For quantitative study, samples of phytoplankton were previously agitated and settled in sedimentation chamber. The cells were counted on an inverted microscope using the intermal method. The first result for phytoplankton density showed a significant variation during study period, with an average concentration of 141.3 times 10 for the power of 4 cell by liter. The result also revealed the presence of two noticeable peaks recorded during summer period. The first one in June and the second one in August. As we can see, both peaks occur in period of warm weather with long hours of sunshine, which concord with many studies. That suggests that phytoplankton proliferation is mostly due to the balance between light and nutrient availability. Qualitative study revealed the presence of 37 species of phytoplankton, belonging mostly to the following 6 phyla: Bacillariophyta, Chlorophyta, Glenophyta, Cyanophyta, Dinophyta, and Chrysophyta. In this second figure, we have some examples for phytoplankton species, in which the most frequent species for diatoms species was Cyclotella ocellata and Aclanthidium catenata. For Chlorophyta, we did have Didymocystis and Clostridium acutum. And last, for Dinophyta, the most frequent species were Pyridinium ombatum. During our study, Hamis Reservoir was mostly co-dominated by Bacillariophyta or diatoms and Chlorophyta, and this all over the study, except for September, where Dinophyta become more numerous. February was the most ascendancy period for Chlorophyta, with 75.4% of dominance, while January encountered the highest share of diatoms, with a total of 72%. For Dinophyta, September was the most ascendancy period, in which this group reaching 45%. The most ascendancy period for Glenophyta was August, except it was not enough to dominate the other groups. The co-domination of Bacillariophyta and Chlorophyta all over the study is mainly caused by the proliferation of Clostridium acuta, which is known by its common adaptation that allow it to grow in low value of light and temperature. The high dominance of diatom can be interpreted as a net growth of phytoplankton that is possibly favored by the low predation and high nutrient availability. The high proliferation of Dinophyta in September was explained in many studies by the fact that Dinophyta has species with high plasticity, including the ability to develop in different reservoir conditions. This good adaptation aptitude is made due to the nature of the cell wall of dinoflagellates. In conclusion, phytoplankton population in Hamis Lake was diversified. The results show a total of 37 species belonging to 6 phyla, in which diatoms represent the most diverse class, covering 18 species, followed by 12 species of Chlorophyta, 9 species of Glenophyta. Samples include as well species from Cyanophyta, Dinophyta, Xophyta phyla. The succession of phytoplankton all over the study were mostly dominated by diatoms and Chlorophyta. Chlorophyta dominance is mostly due to the well adaptation of Clostridium acutum variety, and diatom's dominance is interpreted by the low predation and high nutrient availability in winter. The plankton abundance was particularly marked by 2 peaks recorded during summer, the first one in June and the second one in August. The algae proliferation in summertime period was mostly due to the warm weather for long hours of sunshine. Thank you very much for taking time to attend my presentation.
Thank you so much, Dr. Shafa Amin, for this nice presentation and the nice talk. So, if you have any questions, Dr. Bujnah. No. Any comments for Shafa'i? Okay, thank you so much. Thank you again. So without a delay, we pass sorry to the presentation ID 54. Sorry, ID 11. Lydia Zawi, please share your screen. You didn't send us your presentation. Yes. You can talk, Lydia Zawi. Enable your microphone, please. Lydia Zawi, or write something by chat, please. Lydia Zawi, do you hear me? No. Okay. You can proceed, please, Lydia Zawi. Okay. When you will be ready, Lydia Zawi, you can share your screen. Okay. So without delay, we pass to another or to the next speaker, Bouala Mallouch. Bouala Mallouch. Stop sharing, please, Lydia Zawi. Lydia Zawi. Okay. Bouala Mallouch, are you here? No. Okay. Lydia Zawi, you can start. Okay. Lydia Zawi, do you hear me? Vous m'écoutez, Madame Lilia? We don't hear you. On vous entend pas, Madame Lilia. Lilia, activez votre microphone, s'il vous plaît. Lilia. Vous m'entendez, Lilia? Say yes or no. Right, please write. Okay. I think your presentation is PPT with the narration, I think. Okay. You can proceed. Okay. So we can start. But you have to activate your microphone. Okay. Zawi Lilia, please send us your presentation, okay? Send us on my email. Okay? Because you have a problem in your... No. Okay. So, Bouala Mallouch, no. ID 93. Stop sharing your screen, Zawi. Okay, thank you.
Yes, I'm here. I'm with you.
Okay, Wardiya Saaduddin Zannouch, ID 93. Okay, thank you, Wardiya. 93.
Thank you too.
No,
yeah, I'm here.
Okay,
yeah.
We hear you. Okay, thank you.
My presentation, I sent my presentation.
Yes, I know.
You can share it.
Okay, yes. Thank you.
Okay.
Just for Lilia Zawi, please send us now your presentation, okay?
I didn't see my presentation.
Yes, we have a technical problem.
Okay, okay.
Okay.
Okay, you can start. Can I start now? Why didn't you speak with me? No, no, no. Can I start?
Wait, we have your video, okay?
Ah non. Donc, c'est pas la peine que je fasse la communication. Donc,
Yes, yes.
ah,
Okay. Yeah.
okay, okay. Thank you.
I thought that you send us the video,
Yeah. Yeah.
but you send us this.
It is a video. It's a video, but
No,
I can
it
do it.
doesn't work. Yes, so you share your screen. It doesn't work for us, okay?
Okay.
Okay.
Okay. I'll let me see. I share with you my screen. Huh? Do you see my screen?
Not yet. Yes, your screen, yes. But the presentation, okay, that's good. Thank you.
والله مشكل في البيسي ما حبيت نمشي اروح ولا بدي اروح كاني رايح اروح.
You hear me now?
Yes, yes. Go ahead. Please proceed.
What? Okay.
Proceed. You give up.
Give me some time because I have video in my screen and I can't speak with my video. Okay. Some 2 or 1. Okay. I will delete the. Excuse me for this désagrément.
No problem.
C'est un désagrément. Parce que je vous ai envoyé la vidéo, je croyais que ça allait fonctionner puisque ça ne fonctionne pas autant que ça. Donc, je suis obligée de supprimer ce qu'il y a sur... Parce que ça me dérangerait d'entendre ma voix et de refaire en même temps. Vous voyez?
C'est pas grave. Donc, sans tarder, on passe à l'autre communication. Et quand tu serais prête, vous allez présenter ton travail,
je serai
okay?
dans quelques secondes. Hein, dans quelques secondes. Je vais juste supprimer le...
Okay.
Oui, quelques secondes. À peine... Thank you. Voilà. next one. Okay.
Wait, please. Stop sharing your presentation.
Screen. Ah, d'accord.
We fix the problem now.
The problem is
Okay, thank you.
at your level.
No problem. No problem.
You will share my presentation? It doesn't work?
The quality of the sound is too bad.
It's so low.
Not low, bad.
Bad.
Yes.
Don't. Well, I can do it another time. It's no problem. I can share my screen and do it for the second time.
Okay. So you can do it now?
Why not? Where?
Okay. So please start.
Okay. Est-ce que c'est bon? Vous voyez mon écran?
Yes.
Okay, thank you for this
Because
invitation. I can start now. Thank you for the invitation for the first international conference in environmental technology, biotechnology, and biodiversity. I think that I must introduce myself in first. My name is Desnouch from Béjaïa University. What I'm going to talk about today is the heavy metals concentration in honeybee, Apis mellifera, Demisa forager, pollen, and propolis from 6 apiaries. During the last few decades, pollution by chemicals and heavy metals is accelerated due to mining, smelting, manufacturing, traffic emissions, use of agricultural fertilizers, pesticides, municipal wastes, industrial effluents, and industrial chemicals, etc. Many of these heavy metals are taken up by the plants growing in contaminated soil and show elevated levels in plant tissues as compared to plants grown in controlled soils. Environment matrix analysis is the most direct approach to reveal the heavy metal status in the environment. The honeybee, Apis mellifera, has been investigated as bioindicator of monitored pollutants. And B products can be used as accumulative and reactive indicator. Apicultural matrix analyses such as honey, wax, pollen, or the bee themselves can provide useful indicators of pollution of soil, plant, and air pollution by heavy metals in an area of some square kilometer. The aim of the present study is to verify the effectiveness of bioindicator-based method involving the analysis of honeybee worker, Apis mellifera, intermista, and their products, pollen and propolis, for the determination of the environmental pollution with heavy metals by comparing data obtained by different sampling sets. The biomonitoring study was performed in spring during the year 2080 on 6 sampling sites in Béjaïa. It may be noted that all apiaries belong to particular beekeeper. The site 1, or control apiary, is situated in the middle of nature at an altitude of 500 meters. It contains 10 beehives. The second site is and the third site are situated in Tazmet, are located in Tazmet. The second site contains 60 beehives. It's situated near near train station. The Wed Sahel, so 500 meters far away, a hen house and a picking company. The third set, Apiary 3, contains 60 beehives situated at 150 meters from the highway near an SAR manufacturing soldier. Excuse me. Errant trees and platforms. The fourth set, Apiary 4, contains 15 beehives near a citrus root area and an olive grove in the farming village. See, the fifth set, Apiary is about 10 beehives near the highway number 9, 30 meters from the sea and near the agricultural greenhouse. The sixth set, or Apiary 6, contains 40 beehives in Melbu near the highway 43 in an orchard, fruit trees and olive trees, but also near the wet agrion. Honeybee forager propolized, excuse me, and propolized and pollen were sampled from each beehive during spring to avoid contamination. Plastic gloves were used in sampling and handling propolis and pollen. Bee pollen is collected using pollen traps, device that fit over the entrance at the hive and contain opening just big enough for returning forager to squeeze through. Sample preparation. The pollen propolis and bees were dried for 24 hours at 105 degrees Celsius on drying oven, then annually ground by grinder and packed in porcelain bags. Samples are transferred to a muffin oven at 500 degrees for 10 hours. 0.1 gram of pollen bees and propolis ashes are weighed 5 milliliters of sulfuric acid and 20 milliliters of bee distilled water are added to the ashes. Evaporation and an electric plate at 70 degree until 1/3 of the solution is obtained. 2 filtration were made, the first using 3 micrometer diameter Wattman paper, the second using a searing filter. The solutions are collected in 50 milliliter flasks adjusted with final are transferred in test tube for SAA. In this series of experiments, quantitative determination of heavy metal in honeybee worker and bee products was performed by atomic absorption spectrometer. Results, excuse me, for the concentration of lead, zinc, and copper in bee samples, lead concentration are very low in all of the bee samples analyzed. Values obtained are between 0.001 and 0.69 ppm for an average of 0.223 ppm. These contents are much lower than those determined in various works carried out in the same framework of study around the world. We note these others, and for the zinc, concentrations obtained for all the samples are between 2.408 ppm minimum value for the sample of mildew and 39.244 ppm maximum value for the sample of agricultural village, the average being 9.31 ppm for all the samples of bees. These results are much lower than those obtained in several studies carried out in the same context and which are reported by Lambert 2012. For the copper, results obtained for the copper range from the lowest value of 1.56 ppm to the highest of 3.02 ppm. For an average of 2.3305 ppm. These values are very far from those reported by Van der Steen et al. In 2011, who recorded values between 12.65 ppm and 19.77 ppm. For the concentration of lead, zinc, and copper in pollen sample, lead, as you can see in this figure, the values obtained for lead concentration in the pollen of the 6 apiaries are lower than those obtained by Lambert in 2012. These values are between 0.004 ppm and 0.798 ppm. We can clearly see that lead is present in trace amount only and does not exceed the threshold reported by Lambert. The highest zinc concentration are found in this sample from the two sites of Tezmelt train station and Tezmelt Mizarzor with respectively 7.083 ppm and 7.19 and 5 ppm. The smallest value is obtained for the agricultural village with 1.79 ppm. The average of the zinc concentration in all the pollen samples of the 6 beehives is 3.887 ppm. For copper, as you can see in the figure, this value of copper in pollen samples are between 1.107 ppm and 4.
Sorry for the interruption.
Huh?
Sorry for the interruption. Okay.
Yeah.
That's
Why?
work. Okay, thank you.
It works?
Because here we are in zinc.
On zinc?
Okay.
I'm with on the
yes,
propolis.
The right slide. Okay.
I was no, it wasn't in the right slide. I was in Poland, and there's it was propolis.
But now we are talking about the concentration of zinc.
The concentration of lead, zinc, and copper in propolis sample.
Yes.
Okay.
Yes.
The present values are minimal for lead. We start by lead. The present values are minimal except for the value recorded for the propolis of the apiary of Bacaro, which may be due to the fact that the apiary installed near the highway and that the bee sick the constituent elements of the propolis from wheatbrick trees planted all along the highway. For zinc, it's also present in all the propolis samples with an average of 4.49 and 8 ppm. This content is slightly higher than that found in pollen, which is for 3.88 and 7 ppm. For copper, like you can see in the figure, an average value of 3.378 ppm copper is present in all samples from different localities. However, the maximum value recorded at Mizarzor is met with 8.677 ppm and at Milbu with 7.31 ppm may be the start of an alarm signal. To conclude, data presented in this work are part of the study aimed to verify whether honeybees and their products can be considered reliable biological matrices for the assessment of heavy metal pollution. Specifically, the present work designed to verify whether it could be possible to detect significant variation in the values of heavy metals concentration among samples collected from sampling set characterized by markedly different environmental impacts. For this reason, the collection and analysis of samples refer to freshly produced materials. Finally, our results indicate that the content of heavy metals in finally, our results in that the content of the heavy metals in some collected samples of honeybee workers, pollen, and propolis don't exceed the maximum admitted level of these metals. Therefore, these endings emphasizes the elevated level of environmental pollution with this metal as at such location and the possibility of using honeybee workers and to a lesser extent some of their products like biomonitoring or like biomarkers and indicators of environmental pollution. That's bring me to the end of my presentation. Thank you for listening.
Thank you for your presentation.
Okay, thank
So
you.
again, I ask the same question. If you have any question. Ok, Tahri.
Oui, désolée, j'ai trop aimé votre présentation.
Merci.
Je voulais une petite question, c'est que je suis une bonne consommatrice de miel et pollen et propolis. Donc, ça m'a fait un peu peur le fait qu'il y avait des métaux sur ces produits. Donc, j'ai vu dans votre travail, il y avait la concentration du zinc qui était trop élevée dans le village agricole. À votre avis, c'est dû à quoi?
Il faut rappeler aussi que le zinc est un métal lourd. Généralement, est un métal lourd quand ça accède. Mais ce que vous avez vu sur mes résultats ne dépassait pas les normes, hein, même si c'était élevé. Oui, ça ne dépasse pas les normes. Mais vous savez bien que c'est un élément essentiel dans l'organisme.
Oui, bien sûr, oui.
Oui.
Mais quand il dépasse...
il y a une norme internationale. Quand il la dépasse, certainement, il devient toxique. Comme tous les métaux lourds, vous savez, les métaux lourds, oui. Mais sinon, pour le miel, j'ai déjà fait un travail sur le miel qu'on a.
Mais c'est dû à quoi? C'est dû à quoi une quantité importante du zinc? C'est dû à quoi finalement?
Peut-être c'est dû à la flore mellifère de la région. Parce que c'est le village agricole, il y a donc beaucoup de plantes mellifères aux alentours, ou bien peut-être l'eau.
Est-ce que vous avez fait des études?
Il y a des études, la flore, je n'ai pas fait d'études de la flore, mais il y a des collègues qui ont travaillé sur l'eau. Quelle étude?
Oui, généralement. Pardon.
Oui.
Allez-y, je vous entends.
Généralement, le zinc, il est utilisé dans les peintures. Donc, je ne sais pas, est-ce qu'ils utilisent les peintures dans les plantes pour peindre un peu les plantes, pour éviter les fourmis ou je ne sais pas, parce que par rapport aux océans aussi, au niveau du Méditerranée, on trouve beaucoup plus le zinc dans l'eau.
Oui,
et
justement.
Généralement, c'est dû à la peinture des bateaux antifouling.
J'allais arriver là parce qu'il y a le Oued Sahel qui est à côté aussi. Hein.
Ah, d'accord.
Il y a le Oued Sahel qui est à côté. Donc, il y a des collègues qui travaillent justement sur les métaux dans les eaux des oueds et tout ça. Donc,
oui.
Et justement, il y a une étude qui a révélé qu'il y avait un taux de zinc assez élevé. Bien sûr, l'abeille, elle cherche aussi de l'eau. Et non seulement que l'abeille cherche de l'eau, les plantes aussi, parfois, elles sont, je ne sais pas, parfois, elles sont irriguées avec l'eau des rivières.
Oui, bien sûr.
Et ça, là, on est en train d'émettre des hypothèses, ainsi de suite. Bon, pour l'étude qui a été faite sur l'eau, c'est clair. Nous avons déjà des résultats, mais pour la peinture, vous parlez de la peinture, je ne sais pas, parce que nous, on a récolté on a collecté les échantillons à partir des ruches d'abeilles, ce n'est pas au niveau du verger, donc c'est pour ça. Hein, donc, mais il faudrait savoir une chose, que l'abeille quand même, elle est un bon indicateur, elle peut faire l'affaire parce que déjà on a trouvé dans quelques échantillons de propolis dans une autre étude, les échantillons de pollen aussi, on a trouvé quelques traces de métaux lourds. Mais par contre le miel, je vous assure, je travaille sur le miel aussi, on n'a pas trouvé de traces. Vraiment, le miel il est indemne et donc il est bon à la consommation.
Bonne nouvelle. Donc je n'arrête pas de consommer le miel.
Avec modération.
D'accord, merci, c'est très gentil.
Je vous en prie. Je vous en prie.
Thank you so much.
Thank you. Okay.
No question. Okay, thank you.
Wardia, you can see Wardia. Okay.
Okay, thank you, Wardia.
Thank you so much.
Okay, so we return back to Lilia Zawi. I don't know if you are ready, Lilia.
Hello?
Okay, it's okay for you, Lilia?
Can you hear me?
No. You have to disconnect from the other devices. Okay?
Yes.
No, there is an echo. Okay.
Yes.
You are ready?
You can hear me?
Yes, I hear you very well.
Yes, thank you.
You can start.
Yes. Good morning, everyone. I'm so sorry for the technical problem. I want to present my work, which the name is Assessment of Water Quality Bunamusa River in the Taraf region by Water Quality Index.
Sorry, Madam Zawi. Stop sharing your screen or share your presentation.
I stop it?
No, please mute. Okay. Okay, so we will share your screen, okay? But till now we have the problem of your phone. Okay.
Okay. You can hear me?
Yes, we hear you very well now. It's clear.
Thanks.
Okay.
Well, I'm Dr. Zawi from University Betna 2. I want to present you a part of my research, which name Assessment of Water Quality Bunamusa River in the Taraf region by Water Quality Index. Water Quality Index numerically summarizes the information from multiple water quality parameters into a single value. The single value can be used to compare data from several sites. It can be used to look at trends over time on a single site. Water Quality Index is an effective tool to represent the general water quality at a certain place and time based on physico-chemical parameters. In the present study, the Water Quality Index has been applied to assess the suitability of surface water quality of Bunamusa River for drinking purpose. The located site is situated in the extreme northeast of Algeria, which Tarf city, drained by Bunamusa River. Sampling site. Water samples collection was made for 2 periods, summer and winter, for 9 sites, selected station at Tarif City drained by Bunamusa River. The sampling site listed front on stable space with mineral deviance relying on geographic condition and easy access. The water sample collection physically from a depth of 20 centimeters from the surface of the water, especially where the course of water was high, to acquire great homogeneous samples. Plastic holder of volume greater than 1,500 centimeter square utilized for the sampling of each sample. Sampling, conservation, and transfer of samples to the research laboratory were performed per standard processes, RODI, and with the processes delineated in the standard methods, with the index score were determined for 12 selected parameters shown in Table 1. Table 1 presents the methods used for the chemical analysis of surface water for 12 parameters. The calculation of and formulation of the water quality index involved 4 equations or 4 steps. After this equation calculation, the calculated water quality index value are classified into 5 classes to choose the water quality status as shown in Table 3. The first step to calculate the water quality index is to set specific weight for chemical parameters and their relative importance in drinking water, as shown in Table 1. The highest weight was given to every parameter due to the importance of the role played in water quality than other. And we have Table 2 with the subject standard for drinking water quality and evaluated unit weight of specific parameter. And we have this, the same in the last one. Table 3 presented the classification of surface water quality according to water quality index range, according to Yadav et al. And Ramiknash et al. And we have 5 classes or 5 categories. The table presented here presented for 2 seasons, summer and winter, and the classification according to Yadav and al and Ramiknash in all. And we have in this study the profiles of the obtained results exposed that for the winter season, the calculated water quality index values varied from 32.80 to 65.77 with an average value of 46.76. And it oscillated from 35.86 to 97.46 with an average value of 47.25 in summer. And we have water from almost all the sample sites can be careful as suitable for drinking purpose. In conclusion, we can say significant difference in water quality index value detected over the periods and across position with the maximum value, maximum water quality index values, which poor quality in summer on downstream of the river, while the lowest values of water quality index, which are better quality, were in winter on upstream of river Bunamusa. Generally, the study area in boot season is under excellent to good category. It's recommended to continue monitoring the water of this ecosystem to facilitate the establishment of all levels to supervise and defend the natural resource of the region. And thanks for your attention.
Thank you so much for your presentation.
Thank you for you.
So if you have any question? No question? Okay, thank you again for Leila Zawi for your presentation.
Yeah, thank you. And good luck.
Thank you. Good luck you too. Okay, Buana Mallouche, ID 54. Mallouche, yes, he is. Okay. Mallouche, are you ready?
Oui, salam alaikum.
Okay. Wa alaikum assalam wa barakatuh. Okay. So this presentation presented by Buana Mallouche from Mascara University.
The nanocomposite. We started our work as indicated in the following outline. Bismillah ar-Rahman ar-Rahim. Hello everyone. On the occasion of the first international conference on environmental biotechnology and biodiversity, we are blessed to present our oral communication titled by Synthesis, Characterization and Photocatalytic Degradation of Toxic Pollutant by Zinc Cobaltite, doped with tin oxide nanocomposite. We started our work as indicated in the following outline. First, introduction. Second, synthesis of materials. Third, result and discussion. Fourth, photocatalytic activity. And finally, conclusion. Introduction. The main objective of this research is to synthesize and characterize stable and highly active inorganic materials in the field of catalytic for environmental protection. The chosen synthesis method is critical, and we will then study the structure and texture using various characterization techniques. The materials will then be used as photocatalysts for degradation of toxic methylene blue solutions. Synthesis of materials. Sol-gel is the synthetic process used zinc cobaltite and tin oxide solutions were produced, then mixed and dried at 18 degrees to create a gel, which was sequentially calcined at 415 degrees to get crystalline powder of SnO2, ZnCO2O4. A result and discussion. X-ray diffraction analysis. The X-ray diffraction analysis data reveal that the zinc cobaltite has a cubic spinel structure. The appearance of peaks 110 and 101 corresponding to SnO2 tetragonal structure reflections. Infrared spectroscopy analysis. The findings demonstrate the existence of vibrational bands with wave numbers of 669 and 587 centimeter minus 1 for zinc oxygen and cobalt oxygen respectively. SEM analysis. The particles are irregular in shape and size with a tendency towards spherical shape in the case of ZnCO2O4. See figure A. Irregular shape of SnO2 ZnCO2O4 powder due to the formation of many agglomerates C4C. ADX analysis. The presence of a three constituent element of the synthesized material, namely cobalt, zinc, and oxygen, indicates the formation of ZnCO2O4. Tin is well presented. See figure on the right, where recorded the reduction in the percentage of zinc and cobalt when the SnO2 level is increased. Photocatalytic activity. We discovered that photocatalysts destroy methylene blue when exposed to visible light. The amount of SnO2 is the catalyst effect. Its activity: SnO2 20%, ZnCO2O4 provided the highest methylene blue conversion efficiency. Finally, conclusion. According to the literature, zinc cobaltite and tin dioxide are semiconductors with very remarkable photocatalytic properties. In our work, we have chosen the sol gel process for the elaboration of materials. The rate of SnO2 incorporation significantly changed the properties of ZnCO2O4. By comparison of the photocatalytic degradation results, an improvement in photocatalytic properties was recorded in the case of the SnO2 20% ZnCO2O4 material. Thank you for your attention. Please accept my best regard.
Thank you again, Bwana Mandu. Thank you again, Mr. Bwana, for the short presentation. Do you have any question? Do you have any question? No question? Okay, thank you so much. Okay, thank you so much for the presentation. If you have any question, you can write now by chat or send him an email, okay? Now we move to the ID 101. I think there is Professor Munia Baha who raised her hand.
Non, je me suis trompée, excusez-moi. Merci.
Okay. Thank you so much. Okay. So, Abdelmajid Habshi, are you here? Abdelmajid Habshi, I think no. No. Okay. We go to the presentation ID 112, given by Yamina Tahri. Yamina Tahri, I know that she is here. Okay.
I'm here.
Okay, I know. Let's start her presentation.
Ladies and gentlemen, good morning. Let me introduce myself. My name is Yamina Tahri. I'm a doctor in environmental science. Today I will present my work entitled Assessment of Metal Contamination in Sea Cucumber, Oloturia tuberosa, from the Oran Coast, Algeria. The coastal area is considered to be a very important aquatic system ecologically, supporting a wide variety of plant and animal species. The pollution by various chemical substances of these coastal areas may have serious consequences for their ecological balance. Among the pollutants, the heavy metals are considered as a serious problem because of their toxicity and their ability to be accumulated in the nutrient chain. The heavy metal in the marine environment can be assessed by the concentration in water, sediment, or aquatic organisms. The choice fell on Oloturia tuberosa, which is an important part of the Posidonia oceanica ecosystem. Oloturia tuberosa plays an important role in the recycling of organic matter and the oxygenation of seabed sediment. The Oloturia tuberosa is very common on our Algerian coast. It's sedentary species, easy to collect and easy to identify. The study was conducted on the Oran coast in the northwest of Algeria. 3 sites were selected. The first station is La Madrak. It's located on the west of Cap Falcon and is a rocky site forming a large bay open to the east. Upstream of this coastal site, there are farms that are part of the National Plan of Agricultural Development. The farm may affect the coastal marine ecosystem through the use of pesticides, fertilizer, and phytosanitary products. The second station, Ain Freni, is located between 2 headlands forming the large bay of Oran, Cap Ferrat to the north and Cap Falcon to the southeast, and 8 kilometers far from Christian. This station is considered as a low-impact anthropogenic area, but there is presence of source of sulfuric thermal water. Finally, the third station, Cap Carbon, is located near the agglomeration of Arzo. The study, based on the 12 sampling months from February 2014 to January 2015, from 10 sea cucumber were collected each month by scuba divers at a depth of up to 10 meters. Laboratory, each animal was dissected along the ventral surface and eviscerated. The different compartments as the female gonad and male gonads, calcareous ring, the intestines, and body walls. All these compartments were mineralized according to the method of Amiar et al. 1987. For the statistical analysis, I used the software SPSS 20. For results and discussion, the study of the metal contamination of the different sea cucumber compartments considered is shown in this table. For Aaron,
maximum concentration with 18.84 plus or minus 16.90 ppm fresh weight for Lamadrak, and the second for carbon, and the third for Einfrenin. It is noticed that the highest concentration for copper were recorded at the level of the calcareous ring with 10.20 plus or minus 2.45 ppm fresh weight for Lamadrak. On the other hand, for carbon and Einfrenin, the copper concentrations are at the level of intestine with 11.86 plus or minus 6.89 ppm fresh weight and 8.57 plus or minus 5.10 ppm fresh weight respectively. Concerning the zinc concentration is recorded at the level of female gonads with 18.07 plus or minus 2.12 for Lamadrak. While for the two other sites, carbon and Einfrenin, the highest concentration for zinc are at the level of the calcareous ring with 13.2 plus or minus 2.94 ppm fresh weight and 12.85 plus or minus 4.71 ppm fresh weight respectively. The comparative analysis by the post hoc test of metal concentration show that for copper, the interseed variation is highly significant. However, the interseason variation is not significant between autumn and winter. The interorgans variation is recorded highly significant between female gonads and male gonads, between female gonads and calcareous ring, between female gonads and body walls, and a female gonads between intestine. The intersex variation is recorded as highly significant. The statistical analysis for zinc shows that interseeds, interseason, interorgans, and intersex are recorded highly significant. However, for cadmium, interseed variation is significant between Lamadrak, Einfrenin, and carbon, but not significant between Einfrenin and carbon. Interseason variation is recorded significant between winter and the spring, and between spring and autumn. Interorgans variation is significant between body walls and intestines. The intersex variation is recorded not significant. The statistical analysis for the Iran show that interseed variation is highly significant. However, the interseason variation is not significant between winter and spring. The interorgans variation is recorded not significant between calcareous ring and body wall. In addition, the intersex variation is recorded highly significant. Sorry. For cadmium, the correlation is significant, and the correlation of the cadmium concentration with the different metal considered gives a highly significant positive correlation with iron. Therefore, no correlation is observed between the other metal, zinc and copper. The iron was most accumulated in the calcareous ring and the female gonad, compared to the copper where it was higher in the intestine. The zinc was accumulated much more at the liver calcareous ring and male gonads. The comparison of metal trace of secant camber in different sites around the world. Our result obtained showed a similarity with the result of Warner et al. 26 and Sikuro et al. 2012, July et al. 2016. It was observed that the iron concentration were the highest, while copper was present in low concentration. In conclusion, the study shows that the secant camber in the field of bioindicator and biomonitoring turns out to be interesting. The period and the place of sampling, as well as different compartments of the samples, affect significantly the concentrations of copper, zinc, iron, and cadmium in Olotoya tubulosa. The results show the 4 metals studied were found to be selectively distributed in the different body compartments of secant camber: calcareous ring, body wall, intestine, female gonad, and male gonad. Analysis of secant camber organs reveals significant contamination of essential metals, namely iron, zinc, and copper. Those metals are not toxic to certain tolerance level. On the other hand, a high concentration can create physiological disturbance of the body. Thank you.
Thank you so much for your presentation. Okay. I ask if you have any question.
Yes, I have.
Okay, so Professor Baha has a question.
Yes,
Don't hear you.
Assalamu alaikum.
وعليكم السلام ورحمة الله وبركاته.
I'm very interested for your experimentation and your presentation. Mais je vais parler en français par contre. Poser des questions. Voilà. Alors, moi, quand j'étais petite, je voyais beaucoup des holotures, surtout dans la Madrague et dans la baie en Algérie. Comme je suis une vraie pionnière de la mer, je fréquentais beaucoup la mer, que ce soit l'été ou l'hiver. Et on jouait avec les holotures. Mais maintenant, pour mes étudiants, quand je veux faire un TP, un TD, pour leur montrer les holotures, je n'en retrouve plus. Donc, je me pose la question, où sont ces holotures?
Bon, premièrement, merci pour cette question parce que, en fin de compte, avant, le littoral algérien n'était pas beaucoup pollué. Donc maintenant, vous n'allez pas trouver les oursins et les holoturies.
Oui.
Bon, il faut aller à la profondeur pour échantillonner.
Donc, est-ce que vous pouvez sortir quand même avec cette décision de dire que c'est dû à la pollution?
Premièrement, la pollution, parce que vous savez une chose, la Posidonie n'existe plus au littoral.
Oui,
vous devrez passer jusqu'à 10 mètres,
oui.
vous trouvez.
Oui.
Et la Posidonie, quand on dit holoturie, c'est Posidonie. Ils sont en relation. Ils vivent dans leur...
Oui. Puisque vous êtes certain, donc en sortant de ce congrès, je vais faire des sonnettes d'alarme. Donc, il faut vous préparer pour cette sonnette d'alarme. Donc, je vais vous contacter parce qu'on va sortir des sonnettes d'alarme qu'on va remettre là au niveau du ministère de l'Environnement. Et pour l'holoture, donc, vous pouvez continuer votre travail avec nous pour nous marquer surtout
surtout, c'est très important de nous faire tout un résumé. Est-ce que vous pouvez faire l'historique au moins de 10 ans, 20 ans de la présence des holotures? Je ne sais pas si vous pouvez faire ce travail pour faire ressortir cette sonnette d'alarme, parce que là, c'est très, très important. Okay.
Moi, pour le
donc,
moment, je travaille avec toute une équipe.
J'ai peur que ce rond tarde trop, donc j'ai peur que ce rond tarde trop, donc on va en parler après. Donc, je veux
seulement répondre à une chose aussi. Il y a une pêche, entre guillemets, elle est illégale au niveau de nos côtes. Parce que moi, quand je faisais des échantillons, quel qu'il soit, je trouvais des holothuries. Maintenant, j'ai des amis qui font le même travail que moi, c'est-à-dire moi j'ai terminé ma thèse, il y a d'autres doctorants qui vont faire un autre travail sur les holothuries, et elle m'a dit la même chose. Elle dit je ne trouve plus les holothuries comme avant, parce que moi je lui ai dit avant ils plongeaient pas beaucoup, ils trouvaient quand même, il y avait des holothuries. Maintenant même les oursins, et je vous dis clairement, les oursins je ne trouve plus au niveau de la côte oranaise comme avant. C'est-à-dire avant, au niveau des rochers, je trouvais quelques oursins. Maintenant, non.
Très bien. Enfin, en tous les cas, je vais vous inviter encore une autre fois parce que notre laboratoire va organiser une journée des sonnettes d'alarme des espèces disparues.
Je vais contacter aussi ma directrice de thèse aussi.
Et je redonne tout de suite la parole à Madame Ali. Merci.
Merci pour professeur Baha et merci aussi pour Dr. Yamina Tahri.
Merci beaucoup pour.
So we will continue our conference now with the presentation ID 101, Abdelmajid Habshi. Abdelmajid Habshi. Abdelmajid Habshi, are you here, please? No. Okay. So now the ID Jamila Sadoudi. Jamila Sadoudi. No. Isn't here. Now presentation ID of Mr. Ayman Hajjab or Hajjab. Yes.
Yes, I'm here. Yes, I'm here.
Okay, thank you.
Okay.
And thank you for your patience. Thank you so much.
No problem. Okay, no problem. Can I share my screen or not?
No.
Okay. Donc, je m'excuse.
No, please mute your microphone. Okay.
You hear me now?
Yes, we hear you very well.
Okay.
But we try to share your presentation, okay?
Okay. So thanks for.
No.
Okay. You show my presentation. Okay, I'm sorry.
So please stop sharing, okay?
Okay.
Okay, thank you.
Merci.
I'm happy to present the first international conference on environmental biotechnology and biodiversity. In the title of biomonitoring of groundwater quality based on biointegration of amphipods in northern Algeria. So, first, Algeria is one of the most diverse and understated areas with freshwater fauna. In this part of the world remain partly unknown. This study will focus on the origin, diversity, and distribution of freshwater epigean amphipods in Algeria. It also inhabits various types of freshwater environment, raining water, lakes, groundwater, in which they represent the main invertebrate biomes. The knowledge of freshwater amphipods quite is quite limited and consists mainly of few taxonomic works. The objective of our study is to complete the current knowledge on the diversity and the distribution of freshwater epigens amphipods in northwest Algeria. To do this, we summarize all published works and additional sampling. A comparison of the old and new information with new database allows better understanding of the biogeographic distributions of species in the part of the world. So, this figure represents all parts of an amphipods. We have food, we have antennas, we have the gnathophod, periopods, heliopods, and we have the mesosome, we have metasome, aerosome. So all parts of the amphipods. Study site. We have 169 records in the literature data, and we have 136 new registration in the world. So we have must study site here in Algeria. Materials used. Sampling methods used. Surface water. Amphipods were sampled using a hand-net sampler. 500 micrometer match in all types of freshwater environments, springs, streams, lakes, reservoirs, and ponds. So we have groundwater. The phreatobiological allows the capture of plankton and anything that swims in open water, upper water, and harvesting of part of the fauna. So that is attached to the well walls. Samples were preserved in 96% alcohol in the field. All amphipods we identified two species. Diversity of freshwater amphipods. We have we currently now of 42 species capable of colonizing freshwater. All these species belong to two families, the Camaridae and the Pseudonifaridae. More precisely in four genera for the Gamaridae. Echinogammarus stebing with 13 species. We have the Gammarus fabricus, we represent 7 species, and Schistogammarus martinov for 1 species, and Ripidogammarus with 1 species. So in the Maghreb, the knowledge on freshwater amphipods is rather limited and consists mainly of few taxonomic works. The genus Echinogammarus is the most diverse and divided into 3 groups of species: Echinogammarus brilloni, Echinogammarus pengens, Echinogammarus simoni. So, 42 known species in North Africa. All species mentioned with red color. So, species live here in Algeria. Echinogammarus anondale, Echinogammarus haractis, Echinogammarus reductus, Echinogammarus simone, Echinogammarus tacapensis, and Echinogammarus valedictus. So, in the groundwater species, the Pseudonifargus, with 9 species, and other genera. So, the phylogenetic tree of Echinogamma species in Algeria and Tunisia. All the species live here in Algeria and in Tunisia. Conclusion. So, considering the scarcity of fresh water in North Africa, the diversity is relatively high. 10 morphological species and the last 12 cryptic species have been discovered. The fauna is close to the species of Southern Europe, even if only 5 species are present in both sides of the Mediterranean. The geographical distribution and similarity between species support the establishment of ancient lineage, but also the ongoing species process, especially in Tunisia and Morocco. Algeria is the only country that hosts all 3 groups Echinogammaris in all Mediterranean countries. So I finished my work and thank for other channels. So
Okay, thank you for your presentation.
You are welcome, madam.
Wait. Okay. I don't know if there is or there are questions. Yes. Okay, good. Sorry. Professor Baha will ask you some questions.
Okay.
Okay, just a moment.
Oui, salam alaikum. Voilà, votre travail, c'est vrai qu'il est très original. Il y a peu de chercheurs qui ont travaillé ce travail au niveau de l'Algérie.
Oui.
Et j'espère pour vous une bonne continuation. Par contre, j'aimerais bien savoir qui vous a aidé pour faire l'identification.
Ok, pour l'identification, c'est un travail de M. Piscard au niveau du laboratoire de Rennes.
Pardon, je n'entends pas. Attendez, allez-y. Pardon, allez-y.
J'ai dit que l'identification, c'est un travail de groupe au niveau du laboratoire de Rennes.
Ah d'accord, très bien. Parce que là, je n'avais pas suivi. Est-ce que vous l'avez mis pour votre présentation? Que ça a été fait à Rennes?
Non, je n'ai pas mentionné.
D'accord. Oui, parce que c'est très important là. Donc,
non, c'est des données un peu anciennes. C'est un travail donc de 2 ans avant.
Oui, il n'y a pas de problème. Il n'y a pas de problème.
Okay.
Il n'y a pas de problème. Tout simplement aussi que vous parlez de groupe pour les espèces, pour le genre. Et là,
oui.
Que je n'arrivais pas à vous suivre un petit peu parce que là, bon, je ne sais pas si on peut parler de groupe au niveau des espèces ou bien des sous-espèces. Je n'ai pas compris exactement que vous citiez les groupes, parler du genre puis du groupe. Je ne sais pas si vous pouvez m'expliquer. Parce que là, pour la systématique, pour la taxonomie, bon, allez-y, je vous laisse parler.
Donc pour la systématique, en Algérie, l'Algérie héberge 2 groupes d'amphipodes.
D'accord, attendez, vous avez dit 2 groupes d'amphipodes. Donc les groupes, ce sont des groupes de famille?
Non, oui, groupes de famille, les gammaridés, les pseudomycotes.
Voilà, donc ce ne sont pas des groupes des espèces. D'espèces ou bien de genre. C'est ça ce que je me posais la question. Parce que vous parliez espèce, genre, et puis juste après, vous parlez de groupe. Est-ce que vous m'avez compris?
Ce sont 2 familles. Une famille des
gammas.
Voilà,
c'est des familles. Ok, très bien. C'est ce que je voulais obtenir comme réponse. C'est bon. Parce que là, on me presse parce qu'on n'a pas de temps dans ce groupe. Merci beaucoup et bonne continuation. Et c'est un travail que vous devez continuer. Merci. Au revoir.
Merci beaucoup.
Thank you, Professor Baha, and thank you for Ayman Hiddat.
You are welcome. Merci beaucoup.
The last presentation is given by Salih Hasbiri, ID 191. I think Salih Hasbiri is here.
Yes, Assalamu alaikum, I'm here.
Okay, thank you.
Salih Hasbiri. I'm a PhD graduate in microbiology, and here's my research concerning the wheat, entitled Occurrence of Ochratoxin A in Algerian wheat and its myelin derivatives. Let me start by introducing my work. Contamination of wheat with fungal toxins can occur in the field during storage, processing, and nourishment. Cereals such as wheat, rice, corn, and their derivatives represent a staple food for the population, where its consumption is usually in Algeria in the form of couscous, traditional bread, soup based on shredded wheat, and barley. However, the safety and quality of food is a major concern today. It is necessary to establish studies in order to break down the information needed on the OTA content of Algerian wheat. Here's the problematic and objectives of this study. First, we begin by the study of fungal flora. Then we study the producing power of OTA, then the molecular identification of fungal isolates, and then the extraction and determination of ochratoxin A in samples of wheat and its derivative. Then we conclude our work. Let's begin by the study of the fungal flora. The isolation and enumeration of the molds in the samples were made on a DBRC medium using two techniques. First, we did the direct technique, the direct method for ethan in seeds, and the method of suspension and dilution for the crushed samples. 3 repetitions are performed. Incubation takes place in the dark at 28 degrees for 5 to 7 days. Then we receive these results. The main types of molds developed on wheat grains grown on a DBRC medium, such as it's shown in these pictures. The results of our experience showed that all the samples were contaminated by several types of fungi, such as these pictures or these photos that are shown in this slide. It was also noticed that Aspergillus was dominant among the others. Then we realized the production of OTA by isolates of Aspergillus section. The results are shown on this table. OTA is produced by isolates, O, isolated from wheat, and its derivatives. The OTA produced range from 1.12 to 3,303.23 microgram per kilograms. Later on, we worked on the molecular identification as it's shown here. The molecular identification based on its region of the rRNA and on CAM gene allowed us to establish the phylogenetic tree shown in the next slide. Here's a phylogenetic tree based on sequence analysis of the CAM gene of Aspergillus originated from samples of wheat and its derivatives. The species found belong to these following sections: section circumdati, section flavi, and section nigri, as it's shown in the scheme. Later on, we realized this study that talked about the OTA levels in wheat samples. The extraction was made by the immunoaffinity column using HPLC. The overall result of OTA contamination of wheat samples is presented in the table. It revealed that 27 out of the 39 samples were contaminated with OTA, with amounts ranging between 0.21 and 27.31 microgram per kilogram. This is probably due to the climatic conditions that encourages the growth of toxigenic fungi species such as Aspergillus. Durum and soft wheat were contaminated with OTA at levels of 74% and 58.33% respectively. In our study, the percentage of positive samples was higher than those obtained by several researchers. The results of the contamination of wheat derivatives by OTA showed that in extra fine semolina, the average OTA contents were 2.42 and 8.2 microgram per kilogram, respectively. In addition to that, the average concentrations of this mycotoxin in durum wheat flour and the others, the fractions mainly intended for animal feed, were 4.07, 7.16, and 8.65 microgram per kilogram, respectively. In conclusion, this study consists of 200 samples, 19 of them are wheat stored, and 110 of them its derivatives. Results showed that there has been a contamination in all the samples in which all the species of the Aspergillus were dominant. The molecular study showed that the Aspergillus acaricis, Aspergillus wistergii, Aspergillus elaealis, Aspergillus carbonarius, and Penicillium icelandensis resulted in high rates of OTA production. OTA was detected in 62 out of the 81 samples of wheat and its derivatives. 30 samples, 48.4%, exceeded the standards set by European Community. Wheat and its derivatives consumed in Algeria are contaminated with OTA. Our study encourages the necessity of respecting good ethical agriculture practices throughout the nutritional chain and the establishment of the OTA standard in Algeria. I would like to thank you for allowing me to share my work and study concerning the contamination of wheat and its derivatives by the OTA mycotoxin and its impact on the nourishment safety of the nation. I wish you all a wonderful day. Thank you for your attention and for listening to me.
Thank you so much for the presentation. And for me, it's a good result to stop eating the bread and its derivatives. Okay? You are agree with me?
Yes,
Dr. Zubir?
yes, Dr. Alili, yes. It's a good, good information or good